Thanks a lot, Chris. Thanks a lot, the organizers, for inviting me. It's the first time I come to the IWCLL meeting, and it's the first time I come to Edinburgh, so it's double joy for me to be here. And like 10 years ago, we started in the lab to analyze the epigenome of CLL, and at that time we were thinking about DNA methylation as the main epigenetic mark regulating gene expression. But after like several years of very intense research, we realized that DNA methylation actually was not correlating with gene expression as well as we initially thought. And why is that? Is because the epigenome is actually made. It's a language made out of multiple dialects, and is the combine or the collaboration among different epigenetic marks what actually regulates gene expression. What we learned about DNA methylation was a lot about cell of origin signatures, as Christoph just highlighted. We learned that DNA methylation is a cumulative trait. So it kind of is the storyteller of the entire like lineage history or developmental history, not only of CLL, but also of the cell of origin. So in a way, DNA methylation is the whole book, whereas gene expression is the present moment of the cell, and therefore there's no wonder that the correlation is minor. So the marks that actually correlate the most with gene expression are K4 trimethylation, K4 monomethylation, K27 acetylation, chromatin accessibility, and the three-dimensional structure of the genome. I will tell you today briefly 3 stories related to chromatin, right? In 2 dimensions with the histone modifications, and in 3 dimensions by learning how these 2 meters of DNA that we have in each cell are actually creating a network within the cell nucleus that actually becomes deregulated also in the context of disease development. So the first story was published last year. I will briefly summarize the main findings. Was the analysis of the reference epigenome of CLL, the analysis of all these multiple layers that were integrated to get a glimpse on how the genome works. Because actually the epigenome is like the software that controls the hardware that is the genome. So we did, in very few cases, we did this complete epigenomic profiling, because you can imagine that to do all these layers, I mean, it was very costly. But we used an extended series of patients to analyze ataxic and K27 acetylation, which are marks related to regulatory elements, both like accessibility, where transcription factors bind by ataxic, and the activation of regulatory elements marked by histone acetylation. The first thing we did is, well, how do these marks look like individually? And before interpreting the CLLs, we need to interpret the controls. So where are the controls? And here what we see is that the naive cells, germinal center, B cells, memory B cells, and plasma cells, in every single epigenetic layer, they are telling us that they are different. So the epigenome is, every layer of the epigenome is heavily modulated during normal B cell differentiation. And understanding that is the key to understand what's leukemia specific, as Christoph very nice highlighted in his talk, through the DNA methylation perspective. So basically, when we now have a look at DNA methylation at the CLLs, you see here circled, they all are kind of different for normal B cells overall. We then integrated all these layers of information into an integrative model in which we like things in colors, and we like things simple. So this is something that is quite simple, because all this complex information, a lot of computational biology behind, at the end, lead us to create a genome with colors, and colors representing function. For instance, here we have, in the CLLs, we have a region close to TCF4 in orange and yellow, and these colors highlight the presence of an enhancer. And in this particular case, as it was a very large enhancer, it is called like super enhancer. Here in green, we have elongation, a gene that is being transcribed. In red, we have active promoters. So when we now compare this gene, that is the gene that has been described in CLL to be important, right, for the pathogenesis, as compared to normal B cells have this isoform being expressed, but CLLs express also this isoform, and then activate this mega super enhancer, right, distantly. This enhancer was more open in CLL than in normal B cells, became demethylated, was not expressed, the enhancer was not expressed, but actually led, through a three-dimensional looping of the DNA, led to overexpression of the gene. We did this analysis by comparing CLLs to regions that were stable in normal B cells to identify or possibly identify a CLL signature that was present in all patients at the chromatin level. And indeed we did. We found 500 regions that became de novo active. That means that they were not active in normal B cell differentiation and became active only in CLL, like this super enhancer and many, many others. I believe the most important finding of this was not the target genes of these regulatory regions, but the fact that these regulatory elements were enriched in transcription factor binding sites of very few transcription factors, mostly in FAT, TCF-LEF, and the FOX family. So it seems that few transcription factors may be actually in charge of programming the chromatin landscape of CLL. So if they are so important to create this CLL-ness chromatin signature, they may also be very good therapeutic targets. Another thing we did is we compared the IGVH mutated and unmutated, and we identified clear differential signatures. So these are the IGVH mutated, the IGVH unmutated, and then here there are regions that are more active in this group, in the good prognosis, and these regions more active in the poor prognosis group. But what's the origin of these changes? To know that, we have to know how normal B cells behave. To properly interpret these changes, we need the normal B cells. And the surprise is that when we put here normal B cell differentiation, actually this upper part is a de novo activation in the mutated CLLs, whereas this second part, this second region here, is actually a de novo inactivation in the good prognosis. So you see the importance of linking the patterns we observe in disease, not only to a suspected cell of origin, but to the entire lineage, to identify what is physiological, normal, and where it is disease-specific. We link also chromatin patterns to mutations. We found, surprisingly, that only probably MYD88 and trisomy 12 were related to specific chromatin patterns. In the case of MYD88, it was very nice because the target genes of the regions that became active were like target genes of the NF-kappa B pathway, so all made sense. But in the great majority of genes or genetic lesions, they didn't correlate with gene expression. Why? Well, we didn't know at the time, but after thinking, what's common in MYD88 and trisomy 12? Well, I think these two are more frequently clonal as compared to other mutations. So if you'd find a case with a particular mutation, but it's present only in 30% of the cells, and then you do the analysis of the chromatin, which is in bulk cells, they may not correlate. To do that, and actually this makes sense because this recent paper from Dan Landau's group is saying that the epigenetic profile of different subclones with different mutations may be different. So to properly understand the link between genetics, epigenetics, and transcription, we may end up having, to enter into the single cell field and combine different molecular layers at single cells. The second story highlights the power of collaboration. Groups working in CLL, but in totally different fields. The group of Richard Hulston, a world expert in genetic predisposition to CLL, they have identified over 40 risk loci that predispose to disease, to CLL, and we came with our map of the functions of the genome based on the epigenome. And the question was, well, can chromatin landscape help us to better understand genetic predisposition in CLL? Indeed, when we took the SNPs that predispose to disease, and not only the SNP, but the entire linkage disequilibrium region, right? We saw a huge enrichment in regulatory elements here. You see this part here? These are enhancers. And you see that the great majority of the SNPs, with the exception of very few, are actually enriched in regulatory elements. The important part of the study was as well that some of these SNPs that predispose to disease they have been described because they are in a particular array. And not necessarily they have to be the functional SNPs, right? They are associated with risk, but not necessarily are the functional SNPs. This particular region, this is the SNP that was described to be associated with CLL risk. And you see it's in a region that is gray, which is heterochromatin. It's not regulatory region. But this complete linkage disequilibrium area actually contains huge enhancer that becomes de novo active in CLL and contains many SNPs. So all these SNPs, or some of them, may be the ones that are actually functionally contributing to CLL development. To map that, we did a huge effort with a series of ataxic patients, 100, K27 acetylation patients, DNA methylation almost 500, and gene expression in 450. And in these patients, we had the SNPs. We knew which alleles we have, whether it was risk loci, the risk allele was homozygous or heterozygous, or was the normal allele. And we compared the levels of all these epigenetic marks and gene expression depending on the presence of a particular, of the risk allele or the normal allele. Through that, there is an analysis called quantitative trait loci. We identified the specific SNPs that are actually correlating with chromatin function and gene expression, and we could identify also the potential target genes of all these regions. Something that is, again, very interesting and links this study to the previous one is that when we analyzed the effect at the DNA level of this particular single nucleotide changes, these particular SNPs, we saw that in some regions, this change of a SNP, what did is was an increased affinity towards particular transcription factors or a decreased affinity. But those that increase affinity are actually TCF and FAT and FOX, which are the ones that we had previously identified as inducing a kind of de novo activation in CLL, right? So I think there is a lot here, food for thought and thinking about early phases of disease in terms of predisposition and in terms of how this is leading eventually to changes in chromatin and then gene function. The third study that I will briefly present today has to do with the 3D genome within the nucleus, or the 3D structure within the nucleus. At the one-dimensional level, there is not a single doubt that these genetic changes, the DNA methylation changes, the gene expression changes have had huge impact in our understanding of disease and also in the clinical management, especially the mutations. But the reality is that these 2 meters of DNA that we have in every cell, they are, packed in the cell nucleus, but they are not only structurally packed. They are different levels of compactation that are actually related to and different levels of three-dimensional interactions that are correlated with genome function. Overall, the genome can be divided into 2 compartments, the active and the inactive, right? Heterochromatin, euchromatin. This is a very classic cytology. Applying a technology that's called in situ high-C, we can actually map this in very detail into the active fraction of the genome and the inactive fraction of the genome. We've done this in normal B-cell differentiation, in mantle cell lymphoma and CLL patients, and what we see, if we have a look at the 3-dimensional genome structure in an unsupervised way, the first thing, again, we look at the controls. Before checking how the disease behaves, we look at the controls. Germinal center B cells, plasma cells, memory and naive are very different. So, conclusion, the 3-dimensional structure of the genome is modulated during normal B-cell differentiation, very much. Actually, 30% of the regions change. CLL and MCL, they are different from each other, and they form distinct clusters. So there may be a CLL signature or a 3-dimensional structure that is specific to CLL. 28% of this 3-dimensional folding and interactions changes during normal B-cell differentiation, so this is something that we studied separately. But there is 72% that is stable, that is not changing in the context of a physiological differentiation of the cells. So if we take this 72%, we ask the question, well, can we then identify changes that are CLL specific, that are not occurring in normal B cells? There is almost 350 regions of 100,000 base pairs that change in CLL. Either that they become more active or that they become more inactive. I will show you just one example. These are the regions that are actually CLL specific, that are even not present in mantle cell lymphoma. And one of them, very clear, is EBF1. So EBF1 is a gene that we know is silent in a very, very high proportion of the CLL cases, and in fact belongs to a signature that differentially diagnoses CLL from other, like mantle cell lymphoma or other lymphoproliferative disorders. We had a look at this area from the three-dimensional perspective, and here these are the chromatin states or the functions of the genome I mentioned before, with different colors reflecting different functions. This is a naive B cell, active promoter in EBF1, elongation, enhancers, etc. And these are the three-dimensional interactions between promoter and different enhancers. So in normal B cells, a lot of interactions in naive B cells. In CLL, the gene becomes heterochromatic, and the functional interactions between enhancers and promoters or different enhancers are gone, totally gone. But then this data can also be useful to do something very fancy. It is to have a look or to have a model on how the EBF1 locus is actually creating a three-dimensional structure within the cell nucleus. This is the structure or the model of the EBF locus in naive B cells. This is the EBF locus that forms it like a different domain, you see. The spheres are enhancers, active enhancers. So there is a lot of activation, and there is a kind of relatively open chromatin structure. In CLL, what happens? Well, the whole chromatin becomes packed, and the enhancers are gone. So in a way, there is, the chromatin is, the level of functional interactions is reduced to a minimum, but the chromatin is becoming heterochromatic and closed, right? And this also, we can measure the volume that EBF1 occupies within the cell nucleus, and in CLL, the volume is much, much, much smaller than in normal B cells. With this, I would like to sum up with just 3 take-home messages. So the first one is that the regulatory chromatin landscape is extensively altered in CLL. In CLL as a whole, leading to a signature that relates to CLL-ness, regardless of the heterogeneity of the disease. All seem to have a particular signature. There are also chromatin signatures related to IGVH subtypes, and it seems that all these chromatin changes seem to be mediated just by few transcription factor families, that the great majority of the SNPs associated to CLL or to CLL risk are located in regions that are enriched in regulatory elements, and that CLLs show an altered 3D genome structure within the nucleus, and that DNA blocks, large DNA blocks, change their level of DNA interactions and contain genes related to disease pathogenesis. So this has been the work of many people and many collaborations. I would like to highlight the role of René Beckmann in the first 2 studies. Now René got a junior leader position at the Genome and the CRG Center in Barcelona. Roser Vilarrasa-Blasi did analysis of the 3-dimensional genomes, but here we started to do all this analysis, and computationally this is so demanding that we say we declare ourselves unable to do it as well as we would like to. So we established a collaboration with Marc Martirlenom, that is the group is just dedicated to analyze 3-dimensional genomes from the computational perspective, and Paula Soler Vila, that was the PhD student that worked very closely with Roser from the biology perspective, Paula from the computational perspective, to make this analysis possible. So again, highlighting the power of collaboration as it happened with the group of Richard Hillstone, because we are not experts in predisposition, so they are. So by bringing forces, we really move forward in the field. And thanks a lot to all the funding agencies and to you for your attention, and I'm a bit late. Sorry.
Thank you. A fantastic talk. Thank you very much, Ignacio. We have time for one question. Dr. Halleck.
Sorry.
I try to then ask a short question. Michael Halleck from Cologne. Excellent talk. And the obvious question is, now you introduce another layer of complexity with the 3D and all the regulation, albeit the methylome is already quite complex. So what is your strategy to fuse these things and this complexity into function? So basically, what is in the concert of all these complicated interactions, there is the driver event that drives CLL into a specific function or regulates migration or all these different things.
I'm supposed to give a short answer to
yes,
that.
but I'm asking about the
tough.
strategy. It's clear that you then don't have an answer, but what is your strategy to actually resolve these questions?
There's two ladies here in the audience, Alba and Judith, that just started in the lab, because we thought about it, and it's like, what do we do now? So DNA methylation goes into clinical applications because it's a great biomarker. Function is like many enhancer regions. Do we go to the target genes of the enhancer regions, or do we go to the cause that activates the enhancers, transcription factors? And what's the cause of the transcription factor activation? In fact, BCR signaling, microenvironment. So we are opening a branch in the lab that actually studies the role of transcription factors and microenvironment in actually downstream activating chromatin and leading to a deregulated chromatin landscape in CLL. So that's the short answer of it. Yeah.
Thank you.
Thank you.